Hi everyone, welcome to Module 3, Working with fMRI Data. We're going to be starting off with Section 1, which is the overview/biological basis of fMRI. And today's lesson is going to be about brain activity. Future lessons are going to be visualizing brain activity, interpreting fMRI, and applications of fMRI. So at this point, we've gone through the neuroscience modules, so we know that the neuron is the basis of brain activity. We're also familiar with the neuron structure as well as action potentials. And one really important concept, especially for fMRI, is that different regions of the brain are responsible for different forms of activity or function. So some key questions that we're answering in today's lesson are how brain activity can be measured, why blood flow correlates with brain activity, and what the magnetic properties of brain activity are. So just to kind of review neuronal activity, like I said before, brain activity is based in the neurons. So there's this incoming action potential, vesicles bind to the synaptic cell membrane, neurotransmitters are released across that synaptic gap, which bind to receptors and causes a signal to be sent. So neuronal activity actually fluctuates as the brain engages in different tasks, and this results in different patterns of activity. And these activity patterns reveal that different parts of the brain are responsible for different functions. So when we're doing certain tasks, neurons in corresponding areas of our brain are activated. So like I said before, one of the key questions we want to answer here are, why does blood flow correlate with brain activity? So this is kind of a dense slide, but don't be intimidated. Basically, all neural signaling processes require energy in the form of ATP. And the production of ATP, which occurs in mitochondria, requires oxygen. So when a region of the brain is activated by a task, the neural firing results in a local increased energy requirement. So as the energy is produced to fulfill that requirement, local energy stores in the tissue are consumed by glycolysis, which is what this figure is to the right. So you don't need to know that, just it's glycolysis. So the question here is, how does the oxygen get restored? So the depletion of oxygen signals a reaction in the arterial sphincters upstream of blood capillaries, and this causes a dilation of blood vessels. And the dilation of blood vessels results in increased blood flow, which restores that local oxygen level that was depleted in order to fulfill that energy requirement. So this sequence of processes is called the hemodynamic response to a neural event. So there's different types of signals that can be produced from this blood flow. There's CBF, which is local cerebral blood flow, and this is the change observed when using an injected contrast agent and perfusion-weighted MRI. Which is something that measures the relative cerebral blood volume in comparison to normal white matter. This CBF signal is usually used for measurement of blood flow for studying activation mechanisms, not brain function. The signal that we're interested in with fMRI is the bold signal, and this stands for blood oxygenation level dependent signal. This measures the change in the magnetic field surrounding red blood cells, and it depends on the oxygen state of hemoglobin, which we'll go into more later on. And this is used for functional MRI, which measures functional brain activity, and we'll be talking more about exactly how it does this. So how does this blood flow relate to a measurable signal for fMRI? So like we said, there's a bold signal, which once again stands for blood oxygenation level dependent signal. And the reason why it's called that is due to the differing magnetic properties of oxygen-rich blood and oxygen-poor blood, which relates, like I mentioned before, to the oxygenation state of hemoglobin, which is the protein in blood that transports oxygen. So there's two forms of hemoglobin. There's deoxygenated hemoglobin and oxygenated hemoglobin. So deoxygenated hemoglobin does not bind oxygen, therefore deoxygenated, and it produces a weaker magnetic resonance signal. Oxygenated hemoglobin binds oxygen and produces a stronger magnetic resonance signal, or MR signal. So as brain activation stimulates increased blood flow, the deoxygenated hemoglobin, is replaced with oxygenated hemoglobin, causing an increase in the bold signal. So as a summary, brain activation causes increased O2 consumption, so increased oxygen consumption because of that energy requirement, which increases the local concentration of deoxygenated hemoglobin because it takes the oxygen from the hemoglobin in order to consume it, and that results in inflow of oxygenated blood to kind of refill that depletion of oxygen in the local tissue, and that causes an increase in the bold signal because there's this rush of oxygenated hemoglobin, which, if you remember, has a stronger MR signal. So, as a summary, functional MRI measures the magnetic properties of blood flow indicated by the different forms of hemoglobin that have different magnetic properties and different magnetic resonance signals. And this can be visualized by the bold signal, which increases when blood flow and thus brain activity increases in the active areas of the brain. So in terms of what's next, the next lesson is going to be talking about visualizing brain activity. So more about how this bold signal or the magnetic resonance signal translate to the visual graphs of what we usually think of fMRI as a form of brain imaging. And we'll talk about some other forms of brain imaging as well. So that's that for this lesson. It's a bit confusing, so if you need to go back and look at the graphs again, feel free, and let us know if you have any questions.